As the manager of a football club, you want your team to have the most sporting success it can. For your players to succeed, they need to be on the pitch rather than recovering from injury. The costliest injuries are to the hamstring, resulting in an average 3 games missed by a player while they still get paid. Genetics is your secret weapon. It can explain up to 65% of variance in athlete performance, and you plan to use it to find injury-proof players. Welcome to the genetic basis of stuff and things. The hamstring is a group of 3 skeletal muscles in their connective tissue on the back of your upper leg, which are usually injured when you change directions too quickly. These injuries can cost money from treatment and unplayed matches, and reduce the effectiveness of the team due to player absence. Even worse, it makes a player more likely to be injured again if not properly recovered. Genetics plays a key role in determining an individual's resistance to injury. Genes provide the code for proteins and enzymes that control growth and repair of muscles and connective tissue, and their altered expression can result in different muscle structures and thus resistance to injury. Altered expressions of these genes in the hamstrings can arise due to environmental, mutational, and epigenetic factors. Luckily, you've discovered a paper that you hope will provide alleles that seem to alter people's resistance to hamstring injury. A study by Larruskain et al. Seems to hold exactly what you're looking for. Larruskain and his team collated a list of 37 single nucleotide polymorphisms, or SNPs, that were previously associated with altered risk of muscle injuries, and identified their presence in 107 male Spanish football players. They followed these players over 5 years to observe their hamstring injuries, and categorized them into acute, overuse, severe, or recurrent injury types. With this data, they associated the specific alleles that each player had to a risk of a certain category of hamstring injury. They then created a model, considering various genes and a player's age, to determine what SNPs put a player most at risk of certain hamstring injuries. One gene stood out amongst the 37 SNPs as being significantly associated with injury risk in all 4 categories: a specific matrix metalloproteinase (MMP) gene, specifically MMP3, also known as stromolysin 1. You consulted a reputable scientific source and found out that MMPs are a group of enzymes produced by muscle cells that digest various components of the extracellular matrix, or ECM. This ECM is what makes up a lot of the connective tissue, and so variance in MMP function could affect the strength and resistance to injury of the hamstring connective tissue. MMP3 is particularly able to degrade many types of collagen, the main component of the ECM, and thus weaken the muscle. In wild-type MMP3, this function is carried out to repair damaged connective tissue, as the ECM would need to be broken down before it is rebuilt. As opposed to the wild-type allele, MMP3 variant RS679620 contains a guanine to adenosine transition, non-synonymously mutating a glutamate codon into a lysine. This affects the amino acid interactions that activate the protein, increasing injury risk by 1.79 times per A allele compared to a GG genotype. Further exacerbating the risk caused by mutated MMP3, the variant is in linkage disequilibrium with an enhancer sequence polymorphism that increases MMP3 expression. This means a player with an AA genotype would be almost 4 times more likely to have a hamstring injury than a GG genotyped player. While several other SNPs were associated with specific types of hamstring injury, none were correlated with risk as strongly or as broadly as MMP3. While these other SNPs generally also caused overexpression of proteins involved in maintaining the ECM or skeletal muscles themselves. Your path seems clear. If these alleles put a player at higher risk of injury, surely you screen players for these mutations and exclude them from your team to lessen hamstring injury risk and save money. Lucky for you, Larruskain et al. Used their data about SNPs and their associated injury risk, alongside other information, to predict which players were at higher risk for hamstring injury in a future football season. Their study seems to be one of the first to attempt to validate a predictive model of hamstring injury risk with genetics. Unlucky for you, their model was about as good as guessing. If you compared 2 players, one with a higher injury probability based on their genes, the chance that the more at-risk player got injured was only 52%. They found, despite the connection between mutations and hamstring injury risk, the predictive abilities of such a model were limited. Other studies have since echoed this sentiment about screening for injury risk, regardless of sport or type of injury. For example, while the MMP3G allele seems to reduce the risk of hamstring injury, it may also increase the risk of ACL injury. The overlap of variants caused by different genes and external factors is very high, so any current predictive testing for hamstring injury risk, or any injury risk, is likely of little value. While genetic prediction may be limited, there are even further limitations on simply giving your players better genes. Genetic manipulation and gene doping are banned in sports, so any substances that you could have used to simply improve your existing players' genetics are prohibited. Phenotypes are complex, and even with a sample of 37 SNPs, the multifaceted interactions between genes and environment makes it hard to reduce success and risk in sports to simply numbers. Until a more comprehensive model can be created, you'll just have to enjoy the beautiful game.